I'm here to welcome all of you before we introduce our guests for this evening. My name is Pam Husband, and I am the Executive Director of the Canadian SADS Foundation. And we're really excited to be presenting this series of webinars. We are calling this series SADS Expert Insights webinar series. And so before we start, I'd just like to talk about a few basic rules that I think will help everyone and keep everything running smoothly. Number one, we're going to ask you during the actual presentation itself, if you could mute yourself and also put yourself, take yourself off the video. Sometimes, whether you intend to or not, you end up being a little distracting being there. And so we're just asking you to do that. We also draw attention, most of you are pretty, we've had several years now of people using Zoom, so most of you are very literate with it. And so find the chat, and as the presentation goes along, you can certainly put questions into the chat. We will open up the room after the presentation, and you'll be welcome to ask questions. You can raise your hand. That's under reactions, I think. You can raise your hand. We'll try and answer questions. Well, not me, but our presenter certainly will. And we're just looking forward to a really entertaining and informative evening. So before we begin, I'm really thrilled to introduce our speakers for tonight's webinar. Mariam Butt is with us. Mariam is an MSc student in the Tibbits lab, involved in understanding the genetic and molecular foundations of inherited long QT syndrome. And also with us is Dr. Glen Tibbits, a renowned expert with extensive experience in cardiology, cardiac physiology, and stem cell research, with a rich academic and research background from institutions like McGill University, UCLA, and Simon Fraser University. Glen has significantly contributed to the field of cardiac arrhythmias and cardiomyopathies, and his understanding, he's here to share his understanding with us this evening. So we're looking forward to an engaging and informative webinar, and I will now turn it over to our presenters. Thank you.
Hello, everyone. We are very excited to talk to the SADS community today. I'm Glen Tibbits, the co-director of the Cellular and Regenerative Medicine Center at BC Children's Hospital Research Institute. I am very pleased to introduce one of the impressive grad students in our lab, Mariam Butt. Mariam works in our lab at BC Children's Hospital using stem cell-derived heart cells to understand better about QTS. It's all yours, Mariam.
Hi, everyone. Thank you very much, Pam, for the introduction, and thank you, Glenn. So today we will be going kind of into the scientific background of what I do to study long QT syndrome, specifically through stem cells. Okay, so typically in our lifetime, our heart is going to beat around more than 2.5 billion times. And we can see in this graphic, it can be kind of overwhelming because there are a lot of parameters that need to work synchronously in order to drive proper heart function. Now, clinically, a doctor is going to make sure that each one of these factors is in proper working order. But really, as researchers, what our job is to delve further into each one of these categories and understand what is happening at this level. And so when we ask the question of what causes long QT syndrome, we mainly get to focus on the electrical abilities of the heart and really understand how the long QT syndrome disease state the cells are affected in this state and how we can rescue it further. So the electrocardiogram, or ECG, is something many of you will be familiar with. Either you've had your own conducted, you've seen a family member, or you've just seen it on TV. But why is it a very important diagnostic tool in a clinician's toolbox? Well, the ECG is a visual product of the carefully timed electrical activity in the heart. And oftentimes, this is the first step taken in order to diagnose an arrhythmia. Sorry, 1 second. We see each peak on this ECG is the electrical basis of an important heart function. So first, to be activated within the heart are the atria, and this P wave represents the contraction of the two atria. We have two of them in our heart. Then the electrical stimulus will travel through the heart to its ventricles, and so that they contract and they push out blood to the rest of our body. And this is represented by this long and narrow peak here called the QRS complex. Now, after a contraction, it's really important that our heart relaxes so that it is filled with blood again in order to sustain the next contraction. And that relaxation of the ventricles is represented by this T wave here. Now, all of these segments are very important, but what are we really interested in when we talk about long QT syndrome? Well, as the name implies, we're really interested in this QT interval. And this represents the amount of time it takes the heart from beginning of contraction to the end of relaxation. Now, when this interval, which is typically around 430 milliseconds for males or 450 milliseconds for females, is prolonged, then that is when we see the heart being vulnerable to degenerating into an arrhythmic episode. Now, really, I'm stressing that the heart is electrical, but we've all seen ECGs, we all know this, but really the question that we like to ask is, what is making the heart beat? Now, the heart contains 2 to 3 billion specialized cells called cardiomyocytes. Now, cardiomyocytes are like a typical cell because they have this border surrounding them that we call the plasma membrane. And within the plasma membrane are these integral proteins that are called ion channels. Ion channels conduct an electrical current that allows propagation of an electrical stimulus through the heart by allowing these ions to pass through them, but in a very careful and exact manner. So in this diagram, you can see that each ion channel is responsible for a specific ion going through it. Now, in the heart, in this cardiomyocyte, the key ion channels are sodium channels, and these allow sodium ions to come into the cardiomyocyte. Then we have calcium channels. These allow calcium into the cardiomyocyte. And then we have a group of potassium ion channels, and these allow potassium out of the cardiomyocyte. So the movement of all of these ions through the plasma membrane and in or out of the cardiomyocyte is responsible for different phases of cardiomyocyte stimulation. Now, we began this presentation by talking about how the ECG is the summed electrical activity of our heart. So that's really the organ level electrical activity. And what we'll see on the right of this slide is the cardiac or ventricular action potential. And we can think of this as an ECG that's occurring at the cellular level. And really, to understand an arrhythmia, we must really start at this cellular level to understand how at the organ, at the heart, how changes occur in a disordered state. So let's just try to break it down here, where this action potential shown here is a ventricular action potential, and this is just to differentiate between the different types of action potentials that we'll see within the heart based on the region that we're interested in. Now, when we talk about arrhythmias like long QT syndrome, we're really interested in the ventricular state. So here we'll be talking about the ventricular action potential. The first kind of step you'll see here is called depolarization. Now, this is when we will see sodium through the sodium ion channels come into the cardiomyocyte. And now sodium coming into the cardiomyocyte excites it. And after this initial excitation due to sodium, we see calcium coming into the cardiomyocyte. And this calcium allows that cardiomyocyte to then contract. And after contraction, what we want is for the cardiomyocyte to relax. And this is what we call the repolarization phase of the action potential, where potassium efflux, or potassium leaving the cardiomyocyte, allows it to relax and wait for the next excitation to come along. So what we can appreciate here is that normal heart rhythm is made up of the careful interplay of these ion channels working together in perfect timing at the cellular level to develop the organ level activity that we see. Now, this atrial action potential will generate by cells in the atria, and it covers the action of the P wave that we see on an ECG. And then this ventricular action potential that we just talked about will cover from the QRS complex to the end of the T wave. But really, the next question is, where do these ion channels come from? Because we are interested in their function and how they regulate electrical activity in the heart. Now, since ion channels are proteins, we'll really have to turn to DNA for our answer for this. Now, we all know DNA is the genetic recipe that our cells follow to create proteins, which are required to carry out necessary cell function. And the recipe for all of these proteins is found in segments of DNA called genes. And each gene contains a specific sequence for a specific protein to be made. For example, cardiomyocytes will need to make specific ion channels that we talked about earlier in order to function properly. Now, what we're interested in when we discuss inherited arrhythmias are variants that are found in these key genes that can cause a protein that's supposed to be made to be abnormal and not function as normal. So for example, here, this is showing a potassium ion channel gene, and this letter in red, which was previously an A, has now become a T. And we call this a genetic variant because it is not what is normally supposed to be there. As a result, the functioning protein is no longer acting how it should be. And if you can appreciate that ion channels need to work together in a very synchronized manner, then taking any one of these out of the equation can cause a dysregulation of the electrical conduction in our heart. So that's what we want to show here, where maybe now you're asking, well, can a single letter change in our DNA actually disrupt the electrical conduction that severely? Well, the answer is yes, because you see, we're going now from a regular ventricular action potential to this one that is quite dysregulated if we were to disrupt any one of these ion channels. Now, what is happening to these ion channels in the state of long QT syndrome? Before the holidays, we had Dr. Susan Etheridge on the Let's Talk SADS episode with Dr. Senatani, and they were really discussing the patient management and clinical treatment of long QT syndrome. However, as researchers, our goal is to ask the question, what's going on inside of the cell? Now, currently, there are about 16 different, or upwards of 16 different subtypes of long QT syndrome, but today I'll be covering the 3 major subtypes. Beginning with long QT syndrome type 1. So long QT syndrome type 1 occurs when there is a genetic variant within a gene called KCNQ1. Now, the KCNQ1 gene creates a protein, which is a potassium ion channel. Now, these variants are termed loss of function variants, meaning that a variant to this degree will reduce the amount of potassium ion channel that we're supposed to have within our cardiomyocyte. Because of a loss of this potassium ion channel, it takes longer for these cardiomyocytes to relax, and so we have a reduced repolarization. At the cellular level, this reduced repolarization translates to a prolonged QT interval at the organ level. Now, a prolonged QT interval is can have very severe implications because individuals with a prolonged QT interval have an increased susceptibility of their heart rhythm degenerating into a specific type of arrhythmia called torsade de pointe, or then further into ventricular fibrillation. Now, both of these can be fatal. And so it's very important to understand what's going on here. Similarly, long QT syndrome type 2 can also be described almost in the same breath, because what we're seeing are variants within a gene now called KCNH2. This gene also encodes a potassium ion channel, but the name of this potassium ion channel is hERG. And so again, we see loss of function variants within KCNH2 that cause a decreased amount of the hERG protein being expressed at the plasma membrane of the cardiomyocyte. Less protein at the plasma membrane will decrease the ability of the cardiomyocyte to repolarize. We see it takes a longer time. We have a prolonged QT interval, and then again, the heart rhythm can degenerate into torsade de pointe or ventricular fibrillation. Now, kind of contrasting to this is the third type of long QT syndrome. This subtype of long QT syndrome is caused by variants in a gene called SCN5A. SCN5A encodes a sodium ion channel. Now, if you'll recall at the beginning of the presentation, we need initial sodium to come into our cardiomyocytes in order to excite it. But these variants that are associated with long QT syndrome type 3 are called gain-of-function variants. Now, this means that instead of we have too much sodium coming into the cell, and so this gain-of-function means this channel is doing more than what it should. So it's exciting the cardiomyocyte to a higher degree, and because it's going to take longer now for this cardiomyocyte to come down from that excitation and go back to its relaxed state, we again see a prolonged QT interval, which is dangerous for individuals to have. Now, we know what's going on at a cellular level, but really the next kind of question that we have to answer is, how do we best model these cardiac disorders such as long QT syndrome? Because there are a lot of complex relationships that are involved that regulate a normal or disease state. And so for us to actually really understand what's going on, we need a very relevant model and something that is translatable to humans. So what are our options? Now, instead of using cardiomyocytes for our model, we can use a different human cell type. So this is kind of like where we would use a host cell that doesn't typically express our protein of interest. So this could be one of the potassium ion channels I just talked about, or the sodium ion channels. And what we do is we put this protein into a cell that doesn't normally have this protein. And it's really good for us if we want to understand the biophysical properties of that singular channel and how it acts. But now when we're talking about complex disease modeling, a model like this, which is called a heterologous expression system, doesn't really allow us to appreciate the complex tissue structure of the heart, because there are many other proteins that need to interact with one another. And especially if we're talking about cardiac diseases, we want to understand how these interactions can be changed in a disease state. Okay, so then how about we use the heart of a different mammal? Well, mouse models are a very common model that is used in research, because structurally a mouse's heart is very similar to ours. It has two atria, it has two ventricles. But when we begin to see differences is when we switch over to the physiological side, because while our resting heart rate can be somewhere between the range of 60 to 70 beats per minute, a mouse is going to have a resting heart rate of around 600 beats per minute. And this is because of the differential expression of those key cardiac genes that I talked about. Now, if our goal is really to understand how these genes are regulated and how they instruct a cardiomyocyte to beat, well, it's really important that we have a human equivalent for this model. So, well, if our goal is relevancy, why don't we just skip straight to the human and go to human-derived cells? Well, this would be great, but if there weren't some very major limitations to this model. So isolated heart tissue samples, which can be obtained through a donor or through an individual who is going through a procedure, are quite difficult to obtain and, of course, highly invasive. We cannot go up to a healthy human and ask them for a piece of their heart. Also, when we have these cells in a lab and we try to grow them in a dish, these cells are not going to reproduce. So really, if we start with a limited number of cells, that is all we will have until we have another sample to replenish it. And then further, if these samples are taken from individuals while they are undergoing a procedure such as open heart surgery, then these cells have really seen a lifetime of drug exposure, and they wouldn't best recapitulate what a healthy heart looks like. Now, this is why in labs like ours, we use stem cells, which have the remarkable ability to differentiate into various different subtypes. Now, if you're wondering about any ethical concerns when we use stem cells, then perhaps you may be thinking about human embryonic stem cells. Now, these are the stem cells that are derived from the inner cell mass of a fertilized egg called a human blastocyst. And after these inner mast cells are harvested from the blastocyst, this blastocyst is effectively destroyed. However, that's not what we do in our lab. We use human-induced pluripotent stem cells. Now, the main difference here is that we are beginning with cells that have actually terminally differentiated. Now, these are called somatic cells. These are the cells that are present in our skin as fibroblasts or in our blood as peripheral blood mononuclear cells. And what we can do is reprogram these cells in a manner that Glen likes to say, go back to the future, and have them recapitulate this embryonic state. This was a method that was published by Shinya Yamanaka, and he actually won the Nobel Prize for this, because if what we're effectively doing is taking these cells that don't differentiate into different subtypes, reprogramming them so that they are now virtually identical to a human embryonic stem cell. And what we can do in a research lab is then differentiate these stem cells into very many different cell types. In our lab, of course, we are interested in deriving cardiomyocytes. And so what we have at the end of this process are human-induced pluripotent stem cell-derived cardiomyocytes, which is really amazing for our research. Now, at our Center for Cellular and Regenerative Medicine at BC Children's Hospital Research Institute, we have generated a very robust infrastructure in order to be able to use these HIPSCs and then to further study inherited arrhythmias and cardiomyopathies. So while the first kind of point here that I introduced in the beginning of this presentation was the idea of genetic variants. Now, many variants can cause the same disorder or different disorders. And what we want to do is see how a variant is going to cause this disease state. Now, what I begin with for my research is I start with a dish of healthy stem cells. And what I want to do is introduce a genetic change within them. And I do this using a process called CRISPR. Now, in the CRISPR process, what we do is we deliver a protein that acts like basically DNA cutting scissors. And once it's delivered into the cells, this protein is going to cut the DNA and then rewrite the DNA to encode a variant of interest that I would be wanting to study. The way we tell whether these cells have effectively been cut and their DNA has been rewritten is using a process called fluorescence activated cell sorting, where we know that the cells that are fluorescing bright red are the ones that have been edited. And then from there, it's a process of picking the colonies of these stem cells to make sure that we're getting the clone that has now a genetic variant present within its DNA that wasn't there before this episode, beginning this process. And we can confirm this using a variety of experimental techniques where we can read the DNA and see whether or not our stem cells have this genetic change. Now, these genetic changes, of course, are going to be done in the genes of the key cardiac ion channels because we want to see what that disease state is doing. Now, after a genome editing process, we have a very limited number of stem cells So what we want to be able to do then is expand these stem cells to millions of them so that we have them for future research use and we can keep them. And this is facilitated in our lab through a stem cell technologies robotic unit. Typically, you can ask any graduate student or any researcher that works with stem cells, there's a lot that goes into them. It's quite a delicate work and it requires appropriate technical expertise. These cells must be maintained in a very sterile environment. They must be grown with a lot of, in a very careful manner. And when we are working with stem cells, oftentimes researchers were handling a lot of different stem cell lines that are all different because of their genome. Now, because of this, we can think there's a lot of human error risk, and there's also interoperator variability, where stem cells are going to behave different in my hands compared to Glen's hands. And so to kind of mitigate all of these challenges that arise, what we're very fortunate to have in our lab is the automated robotic iPSC handler. And this was possible for us to acquire through generous donations and the support by Mining for Miracles that we have here at BCCHRI. Now, this robotic stem cell handler is able to maintain our genome edited stem cell lines and also patient-derived lines. So these can be taken, this is from somatic cells taken from a patient who may harbor a genetic variant. And once their cells are reprogrammed, we can also have it handled by our robotic iPSC handler. So really, this streamlines our entire iPSC cell culture practice. So after we've generated millions of iPSCs, both normal and ones that harbor genetic variants, our next step in our pipeline is to differentiate them into cardiomyocytes. So in the last 10 years, researchers in the cardiac field have found very robust ways to differentiate iPSCs into functional cardiomyocytes using the delivery of small molecules. You'll see these names here, but essentially what we're doing is we're taking our stem cells that are both healthy or genome edited or derived from a patient, and we're pushing them through this cardiac differentiation pathway by providing them with special media and small molecules to then, at the end of 7 to 12 days, have a dish that is full of millions of beating of cardiomyocytes. So you can imagine these having these cells to work with is very important in us for us to compare normal cardiomyocytes and these cardiomyocytes that harbor variants within them associated with causing genetic inherited arrhythmias. So how do we study long QT syndrome type 2 in the Tibbits lab? Well, this is a very collaborative effort that part of my research works with Dr. Thomas Clayton from SFU, who is extremely renowned in the field of electrophysiology, as is Dr. Alvin Schreier. They're both researchers that focus on the hERG protein, which I'll get into in just a bit, and also like clinicians like Dr. Sanatani, who really provide us the insights into the clinical aspects where they treat the patients and the variants that they see. So definitely a group effort in order to study an inherited arrhythmia. So I'm a master's student in the Tibbits lab, and my project is investigating a variant that is associated with causing long QT syndrome type 2. Now, again, this variant is going to be found in a gene called KCNH2, which encodes for the hERG protein. Now, normally, the hERG protein, because it's an ion channel, it needs to be present within the plasma membrane of the cardiomyocyte so that it can remove potassium at the end of the action potential in order for the cardiomyocyte to relax after contraction. Now, the specific variant that I am studying, well, what we see in these cardiomyocytes is instead of the hERG protein being expressed at the plasma membrane, they are being retained within the cardiomyocyte. And effectively, if we have less functional hERG that is present at the plasma membrane, well, then we're going to have decreased repolarization or decreased ability of the cardiomyocyte to relax. And at the organ level, at the level of the heart, we'll see a prolonged QT interval because of loss of this hERG. Now, what are ways that I want to use to study whether or not hERG is making it to the plasma membrane? Well, personally, one of my favorite methods, because of the pictures you get out of it, is immunocytochemistry. Immunocytochemistry is a great way to look at protein localization. Now, what's happening here is we have an antibody that is going to bind specifically to our protein of interest. So in my case, I will have an antibody that is going to bind specifically to the hERG protein. And what we can do is, after this antibody has been bound to its protein, we can excite this antibody using a laser, and this antibody is going to fluoresce. And this fluorescence is what we can pick up under a microscope. Now, I'll draw your attention to this panel over here at the top of the screen. It's called WT, which stands for wild type, and this is what we call normal. So these are normal cells expressing normal hERG protein, where hERG are the green dots that you see. Now, you can see if we zoom into this cell that normal hERG is going to be present at the plasma membrane, as we expect, because that's where we want it to work. But now let's take a look at a variant that is associated with causing long QT syndrome type 2. Well, here we see these green dots, and again, we zoom in. Instead of these green dots being present at the plasma membrane, we see that they're actually stuck within the cell. So they're not being expressed in the same way as they should be. And of course, this is a proof of concept for us to study where the hERG protein is being retained and where it's getting effectively lost within the cell. Now, really, this whole time I've been stressing that it's really important for the heart to generate synchronous electrical activity and for them to maintain their regular heart rhythm. So one experiment that we do to assess electrical conduction of our normal and variant harboring human-induced pluripotent stem cell-derived cardiomyocytes is through high-speed optical mapping. Now, in this process, what we have is a layer of typically around 500,000 cardiomyocytes. And what we can do is we can use a dye that will show us what the action potentials of these cardiomyocytes look like under using a high-speed camera and a microscope. And so to the right here, you see 5 action potentials from these cells that we recorded. And what you can appreciate also is that in comparison, we have the normal cardiomyocytes, and in comparison to the variant harboring cardiomyocytes, the action potential is quite prolonged. Now, these are cells that contain, that I genome edited to contain a variant that is associated with causing long QT syndrome type 2. And you see that the action potential duration, as we expect and as we hypothesize, is prolonged in these cells. So this is a very important way for us to functionally categorize the effect of a variant within our cells and see whether the effect we see in an individual is also what we see at the cellular level. Because now with these cells in a dish, we can apply different drug treatments, we can apply and see what is able to rescue these cardiomyocytes from a prolonged action potential. And this can have very many avenues for therapeutic research. So in order for clinical management of a disorder to change and new therapeutic options to be available, we really need to understand what genetic variants are doing in the cell at a molecular level. When studying long QT syndrome, actually, it's becoming more common where an individual with long QT syndrome may actually have a completely normal ECG, where their QT interval is fine. It doesn't look anything out of the ordinary, but it's only until that individual experiences some sort of cardiac event where their cardiologist will genetically screen them to identify a variant. Now, that's important to be translated over to us at a basic research lab to study these variants and see why are they different from other variants and how do they still cause disease in an individual. But our overall goal really is to create this disease in a dish model using human-induced pluripotent stem cells, because not only can they be genome edited, but they can be derived from an individual. And so this opens up the possibility of personalized medicine, where every individual has their own unique genetic background. And by using stem cells in this manner, we're able to pinpoint whether or not a variant is causative in causing a disorder. This can also help us determine whether some individuals are more at risk of a cardiac event in comparison to others, but also whether or not we can find new drugs that are able to rescue this pathogenic phenotype that is associated with a genetic variant. And again, because all of this is done in a dish, there are no adverse reactions that would put an individual at risk. And this is a very important step forward in research that would translate later on into clinical management of individuals. So we really need to understand at the cellular level what's going on to then take the next step forward to understand how we can treat it at the individual level. And of course, this work is not done on our own. We have a huge team, all of our students in our lab and all of our collaborators, and of course, generous funding from Mining for Miracles that allows this research to be possible.